Right. Okay, today is when we're going to have our prokaryote lecture. We're going to go through the first PowerPoint on prokaryotes. And again, anything that we cover in the lecture today is going to be fair game on lecture quiz number 3. We will do a lab activity after this that is going to be where you would find the information on the post-lab quiz that will show up. That's post-lab quiz 2. We start with small stuff where we're still having to use these microscopes. For the next couple of weeks, it's still going to be microscope intense. And this scale here is supposed to show us that bacteria are about the size of organelles inside of eukaryotic cells. I mentioned this last time. Most bacteria, like that mitochondria, are about the relative size as organelles that you would find in a eukaryotic cell. In fact, a lot of the organelles in a eukaryotic cell, like the mitochondria and chloroplast, were at one time their own prokaryotic cell. They got engulfed and instead of broken down as a fuel source, they were put to work. This symbiotic relationship evolved between the prokaryotic cell that was engulfed and the host cell. There are some tiny bacteria that even the smallest bacteria is larger than viruses. We mentioned last semester that viruses aren't even a full cell. They're just fragments of either DNA or RNA, and they may or may not have a little protein coat around them. But this still doesn't show us a decent picture. Bless you. This still doesn't show a decent picture about the relative scale size between prokaryotes and eukaryotic cells. So I grabbed this picture from the internet. And you can see this is a slide where somebody has taken a toothpick. In fact, you can do this today if you have extra time. You can take one of those toothpicks. You can swab the inside of your cheek, just scraping the side of your cheek, and then putting that on a glass slide and putting a drop of water and a cover slip on it, and you'll see these squamous-shaped cells. These are the little fried egg-looking cell. There's the nucleus inside the cell. So these are big eukaryotic cells, and then the darker little dots that we see, those are each prokaryotic cells. So now you can kind of get a visual comparison of how small those prokaryotic cells are in comparison to a much larger eukaryotic cell. And again, we're still talking about these prokaryotic ones that even under high magnification, you can't see you really can't see internal structures very well. So we will end up looking more about, we will end up looking more at the shape of the cell, whether it is spherical or whether it is rod-shaped, or in the case of Spirogyra there, whether it is kind of a spiral-shaped cell, kind of like that. We saw the 3 domains of life last time, and in this lecture we're going to be looking broadly at Archaea and the bacteria domains because they are both comprised out of these prokaryotic cells. Next week when we get into the protists, that's going to be all of the eukaryotic organisms that are not an animal, a plant, or a fungus. So all of the rest of the branches are going to fall under our examples of protists. And we have a bunch of them in the lab. They just came in last week. Putting our 1406 hats on for a second, we remember some general characteristics about prokaryotic cells. So this is going to go for Archaea and bacteria. We're going to see that they have no internal organelles, so no nucleus, none of the other internal compartments. Their chromosome just kind of floats around in that fluid of the cell, the cytoplasm, kind of like a open swimming pool. Generally, the DNA is going to be just one circular loop. Sometimes these prokaryotic cells will have, in addition to this circular loop of DNA that I sometimes described as like a necklace, the cell will have, in addition to this long necklace, these tiny, what they call plasmids. They're just little circular loops of DNA that are more like a keychain, much smaller loop that can hold a few genes on them. We've got a picture in a second. Prokaryotic cells also have a cell wall, and we'll see that there is one substance in the cell wall of bacterial cells that is not found in Archaea, so that's one difference between the two. One similarity between the two is that they both are going to reproduce by this prokaryotic fission, or what we also called binary fission last time, asexual reproduction. And then towards the end of this PowerPoint, we will see that prokaryotic cells have a wide variety of extracting energy and carbon sources from the environment. We Some of them act like plants, some of them act like animals, and some of them do something completely different. Not like plants and not like animals. We'll see examples of all of the metabolic diversity. We've got examples in lab of the Cyanobacteria, the ones that act like plants. This is just a picture of one of those prokaryotic cells, so we can see this rigid cell wall. You can see the fluid inside that I mentioned, just kind of like a swimming pool where you've got the cytoplasm that's the fluid. You got all of the DNA, that circular loop, just kind of condensed in the middle. You've got ribosomes that are floating around, and all of this stuff is just bumping into one another. There's no internal compartments for separate reactions to take place. There are some other structures that you see. Some of these prokaryotic cells will have a flagella, which provides some motility, kind of like an outboard motor on a boat, pushes it in one direction. And then you have these other smaller structures that are called a pilus, or pili for plural. And these are going to allow these cells to reach on and actually grab other bacteria cells. I'll leave it at that for now. Here's that picture that I mentioned of this circular loop. It's a really long circular loop of DNA, and in addition to this big circular loop, there are tiny loops. See over here, these are additional little loops of DNA that we call plasmids, and those usually contain just 2 or 3 genes. But it's a way that these bacteria cells can swap genes with one another, and not even swap the entire genome. They're just swapping fragments of genes. And they can do this with those tiny little structures that, we'll just jump to this picture, they can do that with those tiny little pili that we can see sticking out of cells. Those particular pilus that bump into one another and exchange fragments of their DNA are called sex pilus for obvious reasons. But it would be as if we were just, if you were to shake hands with somebody and share a little bit of your DNA that way. We don't do that, but prokaryotic cells do. Thinking about how these cells reproduce, we saw this in 1406, that prokaryotic cells reproduce by binary fission, or what's called prokaryotic fission. We said it was asexual reproduction, so you didn't need a partner. One cell can do this all by itself. It's going to make an identical copy of itself, so when you're done, you've got 2 identical cells. And from the DNA's perspective, the DNA loves this, right? Because every gene is going to make it into the next cell. So from that selfish gene perspective, the genes love it. They all make it into the next generation. It's easy to do because you don't need a partner, but what's the downside to this asexual reproduction? No variation. Little to no variation. I mean, the only variation would happen is if there was a spelling error. You're trying to make an identical copy and you put the letter in the wrong place. So there are mutations, but those are the only source of variation in asexual reproduction. Sexual reproduction, there's a variation built into that process in multiple places. We don't see that with prokaryotes. And this is just a picture of that prokaryotic fission. Again, a circular loop of DNA. This was more 1406 stuff. It attached to the side of one of those cells, and then it ripped itself apart so that you end up with 2 identical loops of DNA from the parent cell. So more along the lines of 1407 is we want to know the evolutionary history of these 2 groups of cells. And this shows us geologic time at the bottom. Remembering back to our first Unit 1, we said the first prokaryotic cells show up in the Archaean. That was around 3.5 billion years ago. So origin of prokaryotic cells. And then we see lost my arrow there. We see bacteria along one line, and then along this other line, that is going to lead to the Archaea, the other prokaryotic cells. You also see, and I think I mentioned this previously, that we share a more recent common ancestor with Archaea than we do with bacteria. And that's what this graph is also trying to show, that Archaea and Eukarya split right after those, where are we at here? Trying to think. I guess that's still in the Archaean because it's somewhere around 2.5 that those first eukaryotic cells show up. So somewhere around here is when you had that endosymbiosis that was occurring. If I didn't show the slides on endosymbiosis, we will be getting into that when we talk about protist, how the first eukaryotic cells formed and started to engulf other prokaryotic cells. I don't have, actually, give me one second. We've got one back here. Here's our eukaryotic cell that we're going to be talking about. And well, what's nice about this one is that we can see that we've got one of these eukaryotic cells that's got a big nucleus and a bunch of other organelles inside of it. And one of those organelles in particular, the mitochondria that we see all over the place, those would be about the size of an individual prokaryotic cell. And if we look inside those mitochondria, you remember from 1406, they have their own DNA. It's in a circular loop. They have their own ribosomes. They replicate on their own schedule. And what we're looking at is on this graph, I don't have anything to point with, but the lines that we see moving vertically here, these represent what's called lateral gene transfer or horizontal gene transfer, and it's actually highlighting two of these really key endosymbiotic events between bacteria and the eukaryotic cells. It's not labeled here. I tried to put this one additional picture up here because the first arrow is showing when this eukaryotic cell lineage engulfed its first prokaryotic cell, and that was this mitochondria. And the rest, even though this is really a broad graph, you can see there's one lineage of eukaryotic cells that exist today that were part of picking up those mitochondria. But they were not part of this second endosymbiotic event where bacteria also ended up transferring to some of these eukaryotic cells chloroplast. So this lineage is going to lead to green algae and eventually plants because they have chloroplast and mitochondria. We're on this other branch like fungus and animals where we have mitochondria but we don't have the chloroplast. So we'll talk about this evolution of eukaryotic cells. Again, maybe I'm getting too far ahead of myself. But again, we share a more recent common ancestor with the Archaea than we do with bacteria. So in thinking about Archaea and bacteria, I wanted to make some distinctions between the two. Okay, we've seen this sex pilus in which they share fragments of their DNA. That's what we call this horizontal gene transfer. They share those plasmids or little loops of DNA. Getting into the differences between bacteria and Archaea, we may have mentioned this at least once before. Bacteria are going to have a substance in their cell walls called peptidoglycan. It's unique to bacteria. We're not going to find that at all in Archaea. The other thing that we find in bacteria is a circular chromosome, which is also going to be found in Archaea, but unlike Archaea, we're going to see in bacteria there is no histone proteins. Those are those little proteins that the DNA would spool around when it's trying to condense or package itself up. No packaging proteins for these bacteria. They've got the circular loop of DNA, but nothing to wrap it around. Here is our bacteria clade in blue. If you are interested in prokaryotic cells, then perhaps a microbiology class might be one that you would be interested in taking in the future. It looks more closely at the bacteria and the Archaea domains. It would recognize about 5 major clades of bacteria. And this first one, this Proteobacteria, is a really large group of bacteria. The ones that we really see in our lab, the ones that are not harmful really, are going to be the ones that act like plants, the Cyanobacteria, and then some of these gram-positive bacteria that we might find in the yogurt cultures. Here are just pictures of Proteobacteria. We don't have to know the different types of Proteobacteria, but what we need to know is that they all have things in common, like that they lack a nucleus. They have peptidoglycan in their cell walls. They have circular loops of DNA that lack histones. The ones that we see in class, we have Spirogyra, and as their name suggests, we're going to see spiral-shaped cyanobacterial cells that act like plants. And the gram-positive bacteria, we'll see a couple of examples of. Some of the food poisoning, a lot of this is on the food that you eat. The ones that we have in lab are going to be the Cyanobacteria. These are common in pond water around here. In fact, the golf course I was at this weekend had tons of this stuff growing on the surface, and I'm probably going to grab some of it soon. It would make a nice home for some of our microorganisms in there to have something to eat. But if we look at that under the microscope, and we will do this with some of the stuff we grabbed today, we're going to be looking for these chains of cells that some are going to be photosynthetic, that their job is to act like plants. They take in carbon dioxide and they pump out oxygen. If conditions get tough, they might coat themselves in this protein shell that's called an endospore, and this is what they would use to kind of get through any kind of drought or tough conditions. Occasionally you'll see one of these cells that looks a little bit darker. There's something else going on in this particular cell. This is going to be called a heteroassist, and its job is to fix nitrogen rather than fix oxygen. It's going to take in nitrogen gas from the atmosphere and fix it into a form of nitrogen, a molecular form of nitrogen that plants can use. Kind of an ammonia-based fertilizer type thing. So if you ever get to see the root structure of one of those mesquite trees, they're a good example of this symbiotic relationship between plants and bacteria. Plants, you know, they need nitrogen in the ground. That's one of the fertilizers plants use to grow. And all of that nitrogen comes from the atmosphere. But what's interesting is that plants can't use nitrogen when it's in this gas form that you would find there in the atmosphere. But there are bacteria in the soil whose job it is to fix that nitrogen into a usable form that plants can use. And if you can see down in the roots of a lot of these mesquite trees, you'll find these little nodules. And you can cut those nodules open, and you can make a little glass slide just like that we have here. In fact, I might try and find some of these and bring them in now that I'm talking about it. And what you would find under there are just loads of these bacterial cells. These are tiny prokaryotic cells who are just fixing nitrogen for the roots of this plant. And in turn, the plant's going to pay these cells back off later when it grows big and can produce a lot of sugars. It's going to send as many as 25% of those sugars back down into the roots to pay off those bacterial groups that live down there. So, symbiotic relationship. In fact, plants that don't have this relationship with the pockets of bacteria aren't going to grow as fast as the competing plants around them that do. We're going to see that all over the place. Here's an example of those gram-positive bacteria. These are going to be found on a lot of the food that we eat. Many of them are pathogenic. This picture is one that we see here that's coated itself in one of those hard protein capsules. It's trying to disperse, move to a new person, get into some new cells. Food poisoning, like I said, is gram-positive bacteria are responsible for a lot of the different types of food poisoning. So remember to clean your utensils, like the cutting board and knives, cook food properly. The other thing that doesn't have anything to do with cooking food, but you just need to watch out for things in the environment. This is Spirochete, one of these spiral prokaryotic cells, and it's found in the saliva of particular ticks in this region. And they carry in their saliva these bacteria that can be extremely pathogenic. They can carry things like Lyme disease, which is what you're seeing make its way into that person infected in the far picture. Bacteria, like all the ones that we were just looking at, we're living in normal environments, in tick saliva, on the food that we eat, in the soil. When we think about Archaea, these are sometimes considered extreme prokaryotic cells because they're not going to be found in normal environments that we encounter. They, to distinguish them from bacteria, we said that they lack that peptidoglycan in their cell walls. A lot of them have cell walls, they just don't have this particular protein in them. They do have circular loops of DNA, just like all prokaryotic cells do, but these have something in addition. They have those histone proteins, so this circular loop of DNA can be condensed and packaged up, just like our DNA can be condensed and packaged up. So similarity that we have with Archaea that bacteria don't have. I said these were living in extreme environments. We'll see examples of halophiles. Halophiles, that just comes from the word halite, which is the geologic term for salt. You get halite, you crunch it up, and it's salt. And these prokaryotic cells that can live in these extremely high salt concentrations, we'll call them halophiles. Extreme thermophiles, as their name suggests, love these high temperatures. So there's some places like Yellowstone or any of those geologically active areas where there's magma that comes close enough to the surface that it can heat pockets of water to where they're boiling. And normal bacteria can't survive there at all. Certain types of Archaea can thrive there. They love those high temperatures just fine. Then we'll see Methanogens. These are prokaryotic cells that produce methane, and they typically love to live in places where there's no oxygen. So you might have expected these to be common during that early period of the Archean. But once the atmosphere got changed to have a lot of oxygen in it, that kind of made it difficult for these Methanogens to live in places where there was any oxygen. So we find them in places like our digestive tract. You find them in places like my favorite example is now they're actually trying to mine those things. I don't know if I have much room to draw, but there is, wherever it is, somewhere around here there's a landfill. If anybody's ever driven by that, they used to just dig a hole and put all the trash in the hole and cover it up. But there was a bunch of problems with drinking water and things like that. So what they do now is they dig a big hole, and then they line that big hole with what's like a really thick rubber, like a Tupperware container, and they line this hole with this rubber substance, and once they dump all the trash in it, they seal it off with another layer of this rubber, and then they kind of melt those two layers together. So you end up with this underground huge pocket of trash that's all kind of sealed off. And then they'll stick these little pipes in it, and they'll suck all the air out. And what they've created now is this area that is anaerobic. It lacks oxygen, so certain bacteria like these Methanogens can now thrive. And they start to break down the trash. And when they do, they produce methane gas. And that methane gas will come seeping out of those poles, and they burn it. Typically what they do, when they will burn that methane gas and they heat water. And that water creates steam that goes through a pipe and spins a turbine and generates electricity. It sounds, it's interesting, I was having this conversation with my dad the other day. It sounds like it's futuristic stuff that they're doing, but it's really old school steam engine almost. They're using steam to spin a turbine. It's just how they are getting that heat to boil the water to spin the turbine that's interesting. They can get it by this methane gas. You even see this in nuclear power plants. They're using all of that nuclear energy to heat water to spin a turbine to generate electricity. Anyway, Methanogens, you find them in the landfills now. And then acidophiles, those are going to be bacteria that live in places like, once again, those geologically active areas where water gets so hot that it melts down some of the limestone rock. And that has chemicals in it that change the pH that make it difficult for normal bacteria to survive. But Archaea can thrive in these extremely acidic places. Here are pictures of what I'm talking about that might be helpful. You have some areas that, these are areas where salt water on high tide comes in, and then it evaporates and leaves behind these little pockets of extremely high concentrations of salts, very, very salty water that only these certain types of Archaea can survive in. Here are those pools, like I said, in Yellowstone that are close to pockets of magma underneath the surface that are able to heat this water to boiling temperatures and make it difficult for normal bacteria to survive. These are some of those geysers. Again, we've got geologically heated water that is bringing up chemicals in this dissolved rock, and the pH of this water makes it difficult for normal bacteria to survive. And then here we have the cow. We're not really looking at the cow, but we're thinking of the digestive tract of the cow. It is, these cows eat plants, and they can't digest the plant material, but they have bacteria in their gut that can break down that plant material, and they work in these oxygen-free environments. So these, once again, are Methanogens. They produce methane gas that comes out of the back end of the cow too, just like the stuff that comes out of those pipes. If we could somehow catch that and burn that, we could heat water up and spin a turbine with that too. Okay. Not to say that all Archaea live in extreme environments. A few of them have been found in normal environments, oceans, soil, fresh water. Just those are the exceptions to the rule. Here's just a comparison, just to once again compare Archaea and bacteria to things we will talk about next week. So far we haven't seen anything with the nucleus or organelles. So bacteria and Archaea have those things in common. One of the places they differ is peptidoglycan. We find it in bacteria, but it's not present in Archaea. Also, another thing Archaea have in common with us, the other thing we mentioned was histones. We have those in common with Archaea. Bacteria don't have those histones. And then finally, in our comparison of similarities and differences, we have linear chromosomes, but prokaryotic cells all have circular DNA. They may or may not have histones to wrap around, but bacteria and Archaea share that circular loop of DNA. I said we would talk also about how they get different types of nutrition, and this is extremely diverse. The first group that we see here, what we call photoautotrophic bacteria, these are going to be ones that act like plants. We call plants photoautotrophs because they get their energy from sunlight, and their carbon source comes directly from the atmosphere. They just pull it out of the air. So they can get carbon from carbon dioxide, and we talked about this last time. If you're taking tiny carbon dioxide molecules, if you're taking 6 of those and you're ripping the carbon off of each one to build this larger glucose molecule, you're synthesizing something. You're going from small to large. So energy has to come in somewhere anytime you're synthesizing something. And in this case, energy is going to come from sunlight. I just am kind of fascinated about that when we're thinking of photosynthesis, that it really first arose in these prokaryotic cells and hasn't been reinvented. We just see it has been hijacked basically by these other eukaryotic cells. So cyanobacteria started it. We see those same cyanobacteria in green algae and in plants. Now let's look a little bit more quickly at the other 3 types of ways in which prokaryotic cells can get their nutrients and their energy from the environment. The second one that we'll look at is photoheterotrophic. The photo part is the fact that they're getting their energy from the sunlight, but the heterotrophic part means that their carbon source is not just a free carbon source that's pulled from the environment. This carbon source is going to have to be taken from some type of organic compound. So these are going to be breaking down dead cells, dead waste, some type of organic nutrients. They just can't get their carbon from the atmosphere. So photoheterotrophic, some prokaryotic cells are this, which is weird. This is not like animals and it's not like plants. It's something completely different. But then it gets even more weird when we get down to the next one, chemoautotrophs. Chemo just means that it is not getting its energy from sunlight. Let me try that again because I think I mutated my words. Chemoautotrophs do not get their energy from sunlight. Instead, they are going to get it from inorganic substances. And I know this is broad, but think of those, if you've seen those images of those hydrothermal vents at the bottom of the ocean floor, you know there's these pockets of magma that are just coming up and immediately cool and solidify into these columns of rock. And you see around those columns of rock just tons of bacteria. We're talking about the Archaea here. And they're so far down there that sunlight never reaches them. So sunlight is not their energy source. They're getting their energy source from those inorganic molecules that are coming up from those hydrothermal vents. A lot of them are sulfuric acid, those types of molecules that they're using as their energy source. Again, sunlight is not down there. So there's other ways for some of these prokaryotic cells to get their energy source. Chemoautotrophs get their energy from inorganic substances like that sulfuric acid, hydrothermal vents, and they get their carbon from carbon dioxide. So any autotroph, their carbon dioxide is coming, let me try that again. Any autotroph, their carbon source is coming from carbon dioxide. The last one is chemoheterotroph, and these are more like us. We don't get our energy from the sunlight. We get our energy by breaking down organic compounds from that Snicker bar that I ate earlier. And we don't get our carbon source from carbon dioxide. We can't pull our carbon directly from the air. We get our carbon source from, again, some organic substance that we ingested. Maybe those French fries. So prokaryotic cells there are some that are like us too. They have to get their energy and their carbon from some type of carbon source. Not from sunlight, and not from carbon dioxide. Okay. On to recognition of the types of common shapes of bacteria. These are the common shapes. There are either these spherical shaped bacteria that we call cockeye shaped. These typically, sometimes are by themselves or they will exist in chains. chains of these cells are what we call, and this is in your lab handout, Streptococcus would just be a chain of these cocci-shaped prokaryotic cells. There is bacilli-shaped, these rod-shaped prokaryotic cells, and in some of the bacteria, some of those yogurt colonies, you might see chains of these rod-shaped cells. That would be Streptobacillus. And then we also have an example of one of these spiral-shaped prokaryotic cells. Some are pathogenic. The one that we have over there is not. It's plant-like. So we should be able to recognize, if you see on the post-lab quiz, a picture of a spiral-shaped prokaryotic cell, a rod-shaped bacterial cell, or a spherical-shaped bacterial cell. Here is another picture that I just grabbed from the internet. It shows, again, the common types of bacteria that we are likely to see in the yogurt. Either this Lactococcus, which is, again, that's a type of Streptococcus. It's just chains of those spherical cells. And then this Lactobacillus, it's usually just a few chains of those rod-shaped bacterial cells. Here is our last slide. And you may, if you have a steady hand and you don't have the phone that I have, this one, I cannot get a picture. I used to be able to take a picture with my old phone, but this one, it has a couple of different cameras, and anytime I get it close, it tries to switch to the other camera. Maybe there's a way I can fix it, but if you have a camera that's better than mine, you might be able to hold it above the microscope and take a picture that way. And honestly, I don't care if you draw these things, whatever we find today, or if you just take a picture with your phone. You're not going to have to redraw these, but you are going to have to recognize on the post-lab quiz likely multiple choice questions. Post-lab quizzes are not difficult. These are probably the easiest quizzes that we'll have all semester. It's just a reminder of the things that we saw in lab. So I would say if you're not going to be here for one particular lab, I would get the number of somebody that is also in this class that can maybe send you the pictures of what we found in lab that day. Or if not, I can try and send you some email links that will show you the types of things that we've seen so that you can log on and take this quiz. But anyway, this is an example of the types of things that you might be able to take pictures of. And you can see Streptococcus here. These are smaller chains of the cocci-shaped bacteria. These, there's no rod-shaped bacteria here. This is just an example of the types of things that you'll see even under high magnification. So this is maybe a starting point. I know by looking at some of the yogurt in the previous class that the yogurt we have has both these spherical-shaped and rod-shaped bacteria. So I'm hoping we can get a good picture of the rod-shaped bacteria. Okay. Last slide. It looks like I spent our very last slide there introducing once again what we were about to do in lab. So I'll check my time. It looks like if you want a break between here and setting up our lab, you're welcome to stretch your legs or whatever you want to do. But whenever you come back, just to go through the steps of the lab, they're on the board, but I also have this handout, which I'll just make sure that you get. These have the step-by-step instructions, the longer version of what I've written up there on the board. And this is not a page that you're going to have to turn back in. It just makes sure, it's like your scavenger hunt list that makes sure that you see all of the things that we have set up over there. If you have a picture of each of these things, you're welcome, then that's kind of your checklist. You know that you're ready to go through those post-lab quiz questions. That's the other thing that was in my head. Post-lab quizzes, since these are just kind of recognizing the things that we did in lab and what I considered kind of the easier quizzes, I did not make these unlimited attempts. You get 2 attempts on these. So if you can't recognize anything the first time, then you get a second chance to kind of better that. But that's it. So just prepare for those. That's why you would want to get some pictures even if you missed, to get an idea of the stuff that we looked at.